hear me okay? System's working? Good. Now I can tell they are. Thank you. Let's see if the switch over to the computer. Well, while that's happening, I just want to thank you for coming today. It's a pleasure to be here. A lot of the work that I've done with the bees is based on German scientific research done by two gentlemen and their students, Karl von Frisch and Martin Lindauer. And so this is the story I'm telling you is built to a large matter on the German traditions of excellent analyses of the behavior and social life of honeybees. So it's really, feels like I'm bringing a story home. A fresh and expanded story, but back to its roots. Looks like we're switching over. There we go. Brilliant. Yeah. Can we darken it all, the lighting that's on the screen? That'll bring the images up a little, make them a little nicer. Maybe that's not, yeah, there we go. That's good. Keep going. Good, thank you. So, what I'm going to be taking you on today is on a journey or a story to look at bees not living in hives, but out in the wild, in a forest, in a particular forest where I've been working for some 40 years looking at these bees. And so we'll be looking at the lives of these bees as they live in the wild. There we go. Looking at their lives and how they live all on their own. And why they do so, how they do so, and what the effects of this lifestyle is on their ability to survive without any treatments, without any care, to survive all on their own. And I want to use, also, as Jonathan did, into some writings by Wendell Berry. He expressed this idea so nicely that we don't know what we're doing. We've never known what we're doing. And this could be referring to us as beekeepers. He was thinking of it in terms of agriculture, for farming. We've never known what we were doing because we never know what we were undoing. We cannot know what we are doing until we know what nature would be doing if we were doing nothing. And that's really the explanation of why I think it's been valuable to look into nature, to go into the Arnott forest and see what the bees are doing. It's helped me, and I think it's helped all of us understand what we undo in the natural lives of bees when we put them in our hives and keep them in our bee yards. That's not a bad thing, but it is changing their lives. So the theme of this conference is learning from the bees, and that's certainly the theme of my talk with you this morning. So, what is nature doing? Where and what are we undoing? To answer those questions, we're going to go to a part of the United States that's New York State. There's New York City down in the corner. But we're going to go up deep into New York State. It would take about a 5-hour drive by car up to this region. My home base is in Ithaca, New York. That's where Cornell University is. And it's really in the middle of nowhere, which is beautiful for a biologist. Centrally isolated, we call it. And we'll be working in a forest that's south of Ithaca. And this is a mountain range. The Appalachian Mountains run up through here. And so when we leave Ithaca and we go south, we go up into the mountains. If we went north, we would go up into excellent farmland. But it turns out we'll be going south to the mountains, the hills, and that's where the bees live in the wild. There we go. To give you a sense of what this part of the world looks like, I show you these two slides. The climate is hot summers and very cold winters. It goes down below 10 below, 20 below centigrade. And the vegetation is primarily deciduous forest. And this is gives you a sense of what the woods look like, and that's my house, so that's what winter looks like. Next slide. There actually we jumped 2 slides. I need to go back one. Could I do the slide advancing? That'll help me pace things. Thank you. So here we are. This is a Google Earth shot of this whole region. Here's Ithaca, the base of Cayuga Lake, and as I say, north of it is good farmland, and you can see the land is broken up into farms. South of it is almost continuous forest where it is hilly. And we're going to be working, a lot of what I'll be reporting on is what the bees are doing when they're living in the Arnott Forest, south of Ithaca. Here's an image of what this forest looks like when you're up on the top of one of the hills. You look out, you see forests as far as you can see. It's very poor farmland, but it's excellent land for growing hardwood trees, which is the main industry in this part of the world. This Arnott Forest, it's a teaching and research forest owned by the university. It's 17 square kilometers. Now that's not terribly large in itself, but it's surrounded by hundreds of more kilometers of forest. It's just that Cornell owns 17 square kilometers. And the first step that excuse me, can I do the advancing? Getting ahead of the story here. The first part of the story occurred back in August of 1978. And that was a point where I was just starting to get interested in how bees live in the wild. And I thought a very basic question to address to begin with was just how common, how abundant are wild colonies? And of course, this was long before Varroa was introduced to North America. So how common are wild colonies? So I went to the forest. I guess you will need to advance these. Oh, yeah, I'm turned on. There we go, thank you. To find the colonies in the forest, I use a technique called bee hunting, which is a process where you go to a patch of flowers where you find a bee, and using this bee box contraption, you capture bees in the box, and you introduce them to a food source. And then they will load up at that food source, this comb where the sugar water is. They'll load up, and then they'll fly home and then they'll come back. And you are there with the bees, and you see which direction they're flying off, and that will points the direction to their home. And here's a little plot from one of my notebooks that shows the distribution of information you get. All these bees were flying off about in the direction of south-southwest or south-southwest. So you can see pretty good information about direction, and you can label bees with little paint marks and get round trip times, and that will give you an indication how far you need to go to find the tree. And you work your way down that line, the bee line, back to the tree. There we go. The first mapping was done in 1978, and you see it here. Here's the outline of the forest, the Arnott Forest. Scale bar this is a half kilometer here. And you can see at that time, I didn't search over the entire forest. I searched over about 50% of the forest area. I found 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 bee trees in the forest. And so that became clear there was about 18 colonies living in this forest in 1978. And I thought that was interesting. That works out to about 1 wild colony per square kilometer. Store that piece of information away. 1 colony per square kilometer. Okay? And here's what some of these bee trees look like. There's a big white ash here, and the bees their nest entrance was a woodpecker hole here. Here's another one where it was just a rot where some red squirrels had moved in, and then the bees had moved in. I like to take my dog when I go bee hunting. He's a good guy. Next slide, please. There we go. Now you might wonder, how do you actually find these nests, given that the entrances tend to be high off the ground? Well, when you get near the nest, you do have to go do a tree-to-tree search. looking up in the top of the tree. Unfortunately, if we could play the video now, you'll see why it's actually pretty easy to spot the bees and their nest entrance. There we go. Good. Thank you. So that our eyes, of course, are very sensitive to movement. So when you're looking up in the crown of a tree, the top of a tree, there'll be sunlight glittering off these bees and they're moving around. So it's pretty easy to find their nest entrance once you've found the right tree. Okay? Now, what did I learn about these bee tree homes, these natural homes of the bees? Well, one thing I learned is that entrances are high off the ground, 9.8 meters on average, pretty high up. I also learned that the entrances are quite small in these natural homes of the bees, 22 centimeters squared on average, many smaller than that. I also learned that the size of the nest cavities they're occupying is relatively small, only about 40 liters. And those of you that work with Langstroth hives, well, a deep Langstroth 10-frame hive is 42 liters. So the bees in the wild are living in nest cavities, which are typically around the size of one deep Langstroth hive body, not very large. And if you look inside the cavities, you'll find that the walls and the floor are beautifully varnished with propolis. The bees have scraped all the rotting wood that formed the cavity off the walls and coated the walls with this varnish, this resinous varnish, and that makes it antimicrobial. This is the floor. This is a section of the floor of one of these nest cavities. Next slide, please. So, back in 1978, here were the conclusions I could make. Varroa, it was done in 1978. It was about 1 colony per square kilometer. And at that point, I had switched. That was a nice piece of information to have, but I didn't know how important it was going to be. I only realized that this baseline information about the abundance of wild colonies would prove important not until the 1990s. And what happened in the 1990s was the arrival of Varroa in North America. We first discovered Varroa in our hives at Cornell in 1994, and that winter we had very heavy mortality. I think the mites had been in the hives for a couple of years before, but we just hadn't noticed them, hadn't been looking for them. We had about 80% mortality of our colonies that winter, and even in successive winters, because we were still learning at that point how to deal with Varroa. And the general belief then was that all of the wild colonies had been wiped out because there was nobody giving them Varroa treatments. And I thought that too. I thought all the wild colonies up in the Iron Forest were gone. But I finally, suddenly it dawned on me one day, well, actually, Tom, you could go and check that, go and see if that really is true. So that's what I did. Next slide, please. I went back in 2002 to find out if, in fact, in the 10 years since Varroa arrived, that would have been around 1992, all of the colonies had been killed off. So, and I went back in 2002 to bee hunt, and I went back and next slide, please. I used the same techniques in the same time of year, in the same location, and I found 8 colonies. And I thought, wow, okay, so they're not all killed off up in this woods, in this forest. But then I had to be careful. I just think, well, is that because they're resistant to Varroa, or is it because they don't have Varroa? If they're in a remote location, maybe Varroa hadn't gotten out to the woods. So what did I do? Next slide, please. To answer this question of whether these colonies in the Arnott Forest are infested with Varroa, I put up what I call bait hives. These are just old hives. I mount them up on a ledge, up in a tree. It's a 10-frame Langstroth hive body, and I reduce the entrance. So I've got it high off the ground, like the bees like, got the size they normally occupy, a nice little entrance, which they like, and I was able to catch lots of colonies in these bait hives and then check them for Varroa. What did I find? Next slide, please. I captured 11 colonies, and I found that they all contained varroa. So yes, even deep in this forest, these bees have varroa. And I don't have time to explain today, but now it's perfectly clear that varroa gets around very easily. Probably the primary mechanism is when a colony collapses. Bees in the area, those that rob out that colony, pick up the varroa. We've been studying this robbing of dying colonies. Colonies dying from varroa is a very powerful dispersal mechanism for varroa. So I'm not at all amazed to find now varroa in all the colonies in the Untold forest. What I will be amazed by is to ever find a colony in the wild that doesn't have varroa. So they all had varroa. Only one of the 11 had chalk root. None of them had the foul root diseases. So the conclusion was that all these wild colonies have varroa, and yet they're still there. That made the story even richer. They're still there, but they're laden with varroa. What's going on? Next slide, please. Well, so the question was that how did these wild colonies, how were they able to persist without being treated for varroa? Well, as in all matters biological, there's two general parts to the answer to a mystery. One is the role of genetics, and the other is the role of environment. So that plays out here in terms of perhaps these bees have genetic resistance, and perhaps also, or in place of that genetic resistance, they have a good lifestyle. So that's what we're going to be looking at now in this talk. We're first going to look at the question of do they have good genes for varroa resistance, and then we'll also be looking at do they have a good lifestyle. And by lifestyle, I mean things like the colony spacing, nest site, and nest structure. Are there some things about the way they're nested that help them deal with varroa? Next slide, please. Now let's first look at the genes side of things. And for this work, I worked with one of my students, Sasha Mikheyev. He's now at the Okinawa Institute for Science and Technology. That's like the MIT of Japan. And what we were able to do is do what are called whole genome analyses, which is where we can look across the entire genome of a worker bee. And what we wanted to do, and what we did, was we compared the genetics of bees that I had collected back in 1978 to bees that I then collected in 2010. Now, you might wonder, well, where were those bees in 1978? Well, this was, when you do ecological studies, especially with insects, you collect what are called voucher specimens. So every time I found a bee tree colony in the Arnott Forest, or in a bee tree colony in the other woods outside of Ithaca, I would collect 40 worker bees, and I just pin them, put them in the Cornell University insect collection. I had no idea back in the 70s that they would be useful, but it turned out they are very useful. Could need just one of those bees, could send them to Sasha in Japan, and he could extract the DNA, and we could do the entire analysis. So we did that with both the 1978 bees and the 2010 bees. Next slide, please. This is another Google Earth map of the location of all the colonies that were sampled. A bunch of wild colonies living in trees were sampled down in the Arnott Forest, and then in other areas, other forested areas closer to Ithaca. So we had a sample of 32 wild colonies, both in 1978 and 2010. Next slide, please. Here's an illustration that summarizes what we found, and let me explain what this is. This is an analysis of the genetics in just in the mitochondria of these bees. These are the little parts of the cell that are the powerhouses of cells, and they have the interesting feature that they're only inherited maternally. They're only passed on to mother to offspring, and so they just are passed on through the female line. And so this tells us a lot. Tells us that as we look at what this graph is, and I hope you can see these fine lines, these are basically matrilineal or queen genealogies of these colonies. Now, each of the bees sampled in 1978 is represented by an endpoint on this branching diagram that ends with a little blue symbol. You can't read that, but that just indicates the colony. And all of the lines that branches that end with a red color mark at the end are from the 2011 bees. And what you see is that back in 1978, the colonies were drawn from, had many, many matrilines. There were, in fact, 11 different matrilines, or family history groups back in the wild back in 1978. But in 2011, all of these colonies that we sampled were descendants, their queens were all descendants from just three surviving female lines. And that loss of 8 of the 11 female or mother lines is a sign of a genetic bottleneck. And what produces a genetic bottleneck is a huge population crash, a big loss in a population. And so we can infer from this that these bees that I collected in 2011 are the survivors of strong natural selection. They were the survivors of a small portion of the entire wild population that were able to survive Varroa. So there was very strong selection. It looks like maybe there was 80 or 90% of the colonies were killed off in the mid-1990s. But by 2002, the population had recovered because I found as many colonies in 2002 as I did in 1978. Next slide, please. That was a look at the mitochondrial DNA, and that indicated that this population had gone through a bottleneck. If we looked at when we switched to looking at the DNA that's in the nuclear genes, which is where most of the DNA is, you don't see this bottleneck, and that's because evidently what happened when the population was crashing, there were many colonies that were so weakened by Varroa that they were too weak to produce queens. But they were still strong enough to produce some drones. So if you look at the nuclear DNA, you don't see the genetic loss, which means that in the wild today, the colonies they didn't lose much of their genetic diversity, but they did lose a lot of their nuclear DNA, just in their mitochondrial DNA. And what we find is that if you look across the nuclear DNA, that there's 634 sites on the 16 chromosomes. And with this new technology, you can look across each chromosome. You can compare that chromosome's genetics to the old genetics, and you can find places where things have changed radically. And that's a sign of selection, strong natural selection. And there are 634 sites across these chromosomes which have this strong signature of selection. And part of our work now is deciphering what those 634 sites represent. I can't give you a detailed report yet, but what we can say is that half of those sites are involved in development. So there was a strong selection for changes in the development of the bees, and of course development creates the bee. So it's not surprising there's lots of changes in the development. Next slide, please. Well, what are some indications of these changes in development? Well, one thing is the bees now are smaller, a little bit smaller in 2011 than in 1978. Look at this in terms of measuring the width of the head, the old bees, and the modern bees. It's not a big difference, but it's a striking difference. And likewise, if you look at the span between the wing joints, it's smaller again. So our older bees are bigger, slightly bigger than the modern bees. There are also some behavioral changes between the old bees and the modern bees. We know that the modern bees are actually quite good at killing mites, killing the adult mites. They do this by biting the legs off the mites. These bees living in the wild. And if you capture a swarm and put it in a hive, and then you put a screen under the hive, you find that the mites that collect underneath the screen, they're missing their legs. So they're not doing very well. And whereas if I set up colonies and make the same measurements, but I buy the queens from a commercial queen breeder in California or Hawaii or Florida, not many of the mites that fall out through the screen are missing their legs. So these bees living in the wild in the Arnott Forest are really good at killing the mites, partly by biting off the legs of the adult mites. But they're also good at preventing the mites from reproducing because they uncap infested brood cells. Sometimes they're quite sophisticated about this uncapping. Sometimes they'll uncap and remove the pupa that's infested with the developing mites. But they have another trick too, and that's where they just uncap the cell, don't pull out the infested pupa, and just let the mites crawl out. We don't know quite why that works. And so when you open up your hive, or when you see a colony that has this trait, you see these cells of pupae with purple eyes looking up at you. And that means those bees have uncapped those cells. They'll leave them uncapped for several hours, and that disrupts the mite's reproduction. Has anybody seen, going through your hives, has anybody seen that trait here yet? Yeah, a few. So that's popping up here in Europe too. Great. So behavioral changes as well as developmental changes. So what have we seen so far? Well, basic question is, how are the wild colonies able to persist without being treated for varroa? We've seen that they've got genetic changes, they've got good genes for resistance to the bees, and we're still working out the details of all of those, what all of those genetic changes mean for the bees. Another thing that helps them is their lifestyle, good lifestyle. These colonies in the wild, for one thing, they have a wide colony spacing, and I don't have time to go through that, but that means that when one colony crashes, there's much less spread of the mites to other colonies because they're not right crowded together in an apiary where the robbing is facilitated. So there's wide spacing. Remember, they're about a kilometer spacing between the hives. Reduces the spread of mites when a colony does collapse. So colony spacing helps them in the wild. But there's two things that I do want to talk about, two other things, nest site and nest structure. We have time to look at those. Let's look at nest site. Here again, going back into the forest, and these are what our nest sites look like. Bees are living in these trees, relatively small entrances, usually well off the ground. And here's a distribution. I mentioned this before, that on average, or typically, the nest cavity in the wild is around the size of one deep Langstroth hive body. Some of the nests are a little smaller than that, some are larger than that. And there was one exception where the bees were living in a monster cavity. That's a whole separate story. But most of them are down in this range, quite a modest cavity. Well, how does that affect things? How does that influence their ability to resist varroa? Oh, and I want to stress that this distribution is by choice by the bees. And I know that it's not just by chance. That's not the distribution of cavities that exist among trees. If you give the bees a choice between a 10-liter nest box or a 40-liter nest box or a 100-liter nest box, they always choose the 40-liter one. They want that intermediate size. So they're choosing those nest cavities of that size, and it's helping them with varroa. How? Next slide, please. Well, these small nest cavities, as we all know, are going to promote swarming. The small cavities, you get frequent swarming. And frequent swarming, it turns out, lowers the level of varroa in colonies. And we'll talk about how that happens. But first, let me show you that this really is correct. Frequent swarming does indeed lead to fewer varroa. The way I studied this is I did an experiment where I had two groups of colonies, 12 colonies in each group. Now in one group, the colonies all lived in small hives, one deep length rough hive body. In the other group, they all lived in large hives. And these are the sizes of hives that are used for honey production in where I live, four deep hive bodies. And all 24 colonies were established simultaneously in 2012. And it's a little hard to see, but these two groups of colonies, hives, were close together. There was only 60 meters between the two groups. So they were living in the same location, and it's all 12 colonies were headed by queens purchased from a queen breeder in California. So all the queens came from the same source. And they were all set up established simultaneously in 2012 as little colonies, little nucleus colonies. And that summer, I just let them grow. And they grew up and they survived their first winter very nicely. Next slide, please. Thank you. Now, in the summer of 2013, now the colonies were well established. And in this summer, there was considerable swarming among the small colonies. 10 out of 12 of these colonies swarmed, 83%. Only 2 out of these 12 colonies swarmed, 17%. That's a big difference. And that's not surprising because colonies in this situation get crowded, they swarm. Colonies in this situation generally don't get crowded and do not swarm. And what was the effect of that on the varroa? Well, here's records of the counts of the varroa. This is the mites per 300 bees in the large and small hives. The large records for the large are blue, the mean values are indicated by the red line for the small hive. And we start out in July 2012, and the mite counts are really the same for both groups of bees because they were started out as matched colonies. By May 15th, so starting the second summer, they were not different. But gradually, as the summer of 2013 unfolded, the colonies that were living in the large hives, their mite counts kept going up and up and up, whereas the ones in the small hives stayed low. So at the end of the summer, 6.2 mites per 300 bees in the large hive and only 1.1 in the small hive. And many of these colonies in the large hive group showed the signs of high virus levels. They had deformed wings, but not at all in the small hive colonies. So what was going on here? Next slide. Well, first of all, what we saw in the survival of the following winter, 2013 to 2014, at the end of this, going on from this point on, in the large hive group, of the 12 colonies, there were only 2 that survived. And one of these colonies was a very interesting colony. One of those 2 surviving colonies was really good at killing varroa. We were looking at catching varroa in the bottom of these hives on sticky boards, and there again, those mites that we collected there were missing their appendages. So that colony really had a very good trait for dealing with varroa. It killed the varroa nicely. The small hive colonies, almost all of them survived, 10 out of 12. Instead of 2 out of 12, 10 out of 12. So that's a huge difference. And what's going on with these small hives, I forgot what I've quite got on the next slide, so I'll click on it and see if I can see. Well, could we go to the next slide? Yeah, actually go back to that last slide, please. Could we go back to the last slide, please? Thank you. What's going on with these small hive colonies is that they were swarming. And and when a colony swarms, they export a lot of mites because about 2/3 of the bees leave in the swarm. And those bees are carrying out many of the mites. But even more importantly than that, an effective swarming is that there's a broodless period, especially if the colony produces not just a prime swarm, but after swarms. And during that broodless period, all of the mites are exposed to the bees. None of them can be hiding in the brood, and so the bees can kill off the mites very well. So the effective swarming is probably a very important part of the mite control for colonies living in the forest as well as in these small hives. So what we've seen in terms of dealing with the mites, we've seen that they have good genes and they have a good environment in terms of colony spacing and nest site. What about nest structure? Is there something about the nests in the wild that helps the bees deal with the mites? Next slide, please. So we'll go back and look at these nests. Next slide, please. Well, there's a number of things that are different about the nests of the bees living in the wild. One is the size of their cells. They're smaller. This is what, if you buy comb foundation in the United States, and you have the bees draw out the combs, this wall-to-wall dimension here will be 5.4 millimeters. Now, in the wild, where the bees build their combs without foundation, they build the combs just the way they want, that dimension is only 5.2 millimeters. But that turns out to be not enough to actually, to the best of our knowledge, that does not hinder the varroa reproduction. There's some evidence, but it's actually pretty weak, that very small cells can hinder varroa reproduction. But the bees don't have those small cells in the wild. Next slide, please. But what is going on inside that nest structure is the propolis walls and the coating of propolis on the combs. And the evidence of how this is helping the bees comes not from my laboratory, but from the work in the laboratory of Professor Marla Spivak at the University of Minnesota. Next slide, please. And what they've done is they have set up colonies where in half of the hives, they put in a material which has lots of little slots in it, and the bees fill those slots with propolis, and it creates a kind of propolis shroud inside the hive. And when they then look at the patterns, the level of gene expression for 6 antimicrobial peptides in the bees, between bees that live in a hive that looks like this versus in a hive that doesn't have the propolis all over the walls, they find that the effect of the propolis envelope is it turns up the gene expression, it lowers the gene expression for the production of these antimicrobial peptides. In other words, the bees are not working as hard to fight infections in the hives that have the propolis coating on the walls. So they conclude that this propolis envelope functions as an antimicrobial shroud. I really like that image of a shroud of antimicrobial material. And it's true, and it's accurate. Shroud, antimicrobial shroud. So that's another thing the bees have going for them in the wild. They're not having to work as hard at fighting diseases. And that translates into greater health, which probably translates into the bees having more time to bite off the legs and throw out varroa. Next slide, please. So, what are the consequences of living in small cavities with much propolis on the walls? Well, we've seen that they have small brood nests and small colonies. We've also seen that they have more colony reproduction through swarming and drone rearing, and they have less disease stress, evidently from this propolis, particularly from the propolis wall. So this nest structure, just the way the cavities that they're living in, helps them deal with varroa in multiple ways. Next slide, please. So the point I want to make with this slide is the answer to this question: how are these wild colonies able to persist without being treated for varroa? They have a lot of things going for them. They've got good bees with good genes, bees that have gone through strong natural selection to have resistance, behaviors of resistance to the mites. But in addition to that, it's their lifestyle. They have a good lifestyle. Their colonies are widely spaced, so they don't distribute, the mites don't get cast from one colony to another as easily, especially when a colony crashes. It doesn't, there are not a lot of colonies nearby to pick up those mites. But also the nest site favors resistance to the mites in terms of the size of the cavity and also the nest structure with the propolis walls, especially. Last slide, I think, is this. So this summarizes what I've shown you this morning. These colonies of European honeybees can survive without chemical treatments for varroa, especially up there in the Arnott Forest. But I don't think the Arnott Forest is at all special. I think it's any place where bees are exposed to natural selection. We've also seen that the colonies in the wild have experienced strong natural selection in the last 20 years, probably, almost certainly due to varroa. Very powerful selection due to varroa. And another conclusion is how we keep bees is not how they live in nature. It's not entirely bee-friendly. And this is so by looking to nature, we've learned in a number of ways in which the bees live differently in nature, and also that these ways, some of these differences help them deal with varroa. In the wild, they're not crowded into apiaries. And when colonies are crowded into apiaries, there's easy spread of diseases. In the wild, they're not housed in large hives. They're choosing small nest cavities. But when colonies are housed in large hives, they have greater honey production because they have more storage space, but they have less swarming, and they have poorer colony health. And a lot of that poorer colony health comes from the fact that they have lots and lots of brood, and it's continuous during the summer. So that gives varroa lots of opportunities to reproduce. So these large hives make for poor health, especially with respect to dealing with varroa. And thirdly, our hives, our manufactured hives, come with nice smooth walls on the interior, and that discourages the bees from coating their nest walls with propolis. And so the bees don't make much use of their natural defense of propolis against microbial infections. So this point I'd like you to take home from all of this is bees living in the wild live different lives, and they live lives that differ in many ways. There's no one single silver bullet for how these bees are dealing with varroa. It's a lot of different things. It's who they are genetically, and it's how they live. Colonies are widely spaced, small nest cavities, frequent swarming, rough walls, a lot of things. Now, the good part of this whole story, and I'll just end it at this funny little slide. Last slide, please. Oh yeah, these suggestions. The good part of this is we can adapt what we've learned from the bees in the wild to our beekeeping, even treatment-free beekeeping. And I suggest if you're in a place where you've got wild colonies living around you, capture swarms from those places to get bees with genetic resistance to varroa, get bees that have been exposed to natural selection to have resistance to varroa. I don't know if that's common here in Germany or not, but I bet there are some places. Secondly, disperse your hives. That'll slow down the spread of disease, especially mites. Again, I know Germany is a crowded country, so probably it's not possible for most of you to spread out your hives to a great extent. But if that happens to be possible, that helps. Here's one any beekeeper can do, and that is house your colonies in small hives and allow them to swarm. Now I know probably swarming is a problem in a crowded country too, but just have to maybe be on top of that. The bees will give you less honey, but also much lower mite levels. Or you could artificially, you could split your colonies, do an artificial swarming operation. That works really well to control the mites as well. Install propolis collection material on the inner walls of your hives, and that'll simulate the building of this propolis shroud. That helps the bees fight off infection so they can work hard on other problems. Rear queens from your survivor colonies. Maintain locally adapted stock. Keep in mind that example, that experiment where you set up 12 colonies in large hives, and I expected them to all die from varroa, but there was one colony, and that colony lived for 5 more years without any treatment from me, and it was a very defensive colony, I should say, but it was very good at biting. Those bees were really good at killing varroa, and so I bet some of you will find those such colonies. So rear queens from your survivor colonies. And then give these survivor colonies 1 frame of drone comb per hive body to help them produce lots of drones and spread those good genes. So these are just some suggestions of how to make use of what we've learned about bees in the wild. I wish I had an opportunity to give a second talk. It's called Darwinian Beekeeping. I could go in greater detail about these things. But if you'd like to learn more about the story that I've told you about how bees are living in the wild, I have written a little book called The Lives of Bees, the Untold Story of Honeybees in the Wild. And I am working on a book called Darwinian Beekeeping to explore the practical applications. And the next slide is my last slide. And I just wanted to thank you for your interest and attention and your invitation to speak. I always like coming back to the homeland of Karl von Frisch and Martin Lindauer. Thank you very much.